at these conferences, and pleased to see some of my investors are here. Albert Yuen from HRN. So I'm CEO and co-founder of Thrive Bioscience, and we provide imaging, analytics, and automation for better drug discovery. And I've added another line there, better medicine, because we received an order for 55 instruments for use in vitro fertilization. And this has pulled us into the medical field much faster than we ever expected. Our main market has been preclinical research and mostly drug discovery. So what we do is automated live cell imaging and analytics. And these are examples of our instruments and screenshots. Just like in my background, I've combined many fields. We're doing that at Thrive as well. Our instruments provide automated imaging that enables improved processes, breakthrough insights, and delivery of cell therapeutics and improved procedures. I like to refer to what the chairman of the biochemistry department said, who's one of our customers at a leading medical school. Current tools are failing us because they hurt the cells, only give us 2D single point in time images, and don't capture contextual data or enough data. After using our instrument in COVID-19 research studying SARS-CoV-2 virus, the chairman said, Thrive will change the world. Thrive allows us to understand and see living dynamic cells and tissues in 3D over time with remarkable images. So a little background on Thrive. We have an integrated family of products. We're not a single product family, and they don't require FDA approval. The recent order that's for IVF requires a higher standard of manufacturing, not too different from what we already have, but none of our products require FDA approval, which is different than most of my career and very welcome because it's a risk reduction. We have 75 units that have been purchased and ordered, and we have major collaborations in big pharma and with research institutes such as Harvard Stem Cell Institute, Wake Forest Institute for Regenerative Medicine, and others. We're projecting 4.4 million in sales this year. We've booked 3 million so far, aside from the order for 50 instruments. There's very limited innovation in the field of automated live cell imaging. And so we've been able to file an immense amount of IP to protect ourselves. We have 86 patent applications, of which 38 have issued. We've raised $35.4 million to date, mostly from family offices, the largest of which is the Estee Lauder family, and 7 angel groups, largest of which is Life Science Angels. So we're finishing a Series B convertible note. We're actually in our pro rata to our current investors. If anybody was really interested, there's a chance to sneak in. It's at a very attractive discount of 30%, which is quite investor friendly. Our focus is now on acquisition financing, $5 million of debt with Lawrence and Thrive, for us to acquire a cell culture company that's a leader in environmental controls, which is very important, as I'll explain later. And there's a huge overlap in their $4 million of revenue with our market, and they're a vendor of ours as well. And then towards the end of the year, we plan to do a Series B for 15 million. So as Marty mentioned, I have started, this is my 18th company. I've had 7 unicorns, 3 in software and 4 in life sciences. And Thrive is going to do it again, I believe. And the reason is because of the following strategies. So we're solving a major problem. We're providing a full solution. I read so many business plans where a company is solving just a slice of what needs to be solved, and therefore the full problem never gets solved. We plan to have dominant market share. 2 of my previous companies, Cytic has 90% market share of cytology labs, which primarily read pap smears, and HNC software, to this day, 90% of credit card, debit cards are monitored with my software for fraud. And we plan to do a similar thing, mostly by use of data and a full suite of products. We're not counting on discoveries. Instead, we are competing with cleverly putting together protectable, patentable, existing technologies, again, as a risk mitigation. And we're doing it with a great team, and I always have an eye on the exit. So the problem that we're solving is an important, ignored, inefficient area. Important because cell culture is key to drug discovery, biomedical research, and delivery of medicine, and ignored in that it is still conducted manually as it was 70 years ago, 70. As you can see in these pictures on the left, cell culture plates are kept in these incubators. They're just stacked. Somebody has to reach in, takes them over to a microscope. A few images are taken, a few notes are taken. That's the case in about 97% of labs. Cells cross multiple environments and then are put back in the incubator. Cell culture is a dirty little secret in life sciences. There's huge variability across researchers, across labs, across time that confounds the data. We have contamination rates of up to 11%. Mislabeling of cell lines was at a minimum of 11% and up to 30% in 35 labs that was surveyed by the FDA. And researchers spend hours per day looking through a microscope. And guess what? There's almost no data that's retained. Most apps on our phone, such as Uber, know more about us than we know about the cells we're studying for medicine. This is an expensive problem too. We then spend billions of dollars on these cells of unknown, often poor quality, and that has led to the reproducibility crisis. Nature magazine did a poll of 1,500 scientists, and more than half said that this is a significant problem. Amgen did a study of 53, tried to duplicate 53 studies over 10 years, had between 10 and 100 scientists, and they were only able to replicate 10%. Even if you assume you could replicate 50%, $28 billion a year in the US is being wasted. So you want a solvable problem, and that's what we're doing. We're basically industrializing biology. And we're doing it from a manual craft with very little environmental controls, very little data, not scalable. And with our instruments, combining robotics, fluidics, machine learning, optics, image processing, we're creating more than genomics quantities of data that are automatically captured. So we're getting the right data, enough data. It allows breakthrough insights, it improves processes, and it enables automation. I mentioned that you need to provide a full solution in order to have a big company and certainly a unicorn. And that's what we're doing. In order to solve automated lifestyle imaging, you have to control the environment. You have to have tools and documentations. We have a full barcode documentation system. That's to prevent the mislabeling and the other errors I was just talking about. And obviously you have to acquire very high quality images, which we do. And I'll tell you more about that. And it's important that we automate the imaging. The imaging is done the same way every time. So you have comparable data sets. And of course, those data sets have to be available in a database for analysis. So this is our principal products now and over the next 3 years. On the left is the Cell Assist, which is a benchtop imaging system for just over 100,000. Bottom left, we've combined that with a 50-plate incubator particularly for time series data. Remember, biology is a process over time. Most of our focus now is on software modules to make the system easier to use, to expand the market. Of our 10 engineers, 9, in fact, are software engineers. And most of these programs are using machine learning basically to bridge the problem, which is that biology has tremendous variations. And so to think that a single product is going to solve and make decisions for people is not realistic unless there's some machine learning or user-guided assistance. We're bringing out a low-end imager. And then on the bottom right, we have alpha versions of a $1 million full walk-away automation system that does cell culture, stem cell culture, tissue culture. We have advance orders for 24 of those instruments. So you want to dominate the markets. Um, Two of my companies have had natural monopolies. In other words, that they were monopolies defined by the FTC, but they were obtained not through being predatorial, but because of the quality of the products. So products to succeed, I believe, have to be at least an order of magnitude better. Otherwise, people aren't going to switch. And we have the advantage that our products are actually applicable to all aspects of life sciences, all the different fields. Stem cells, infectious disease research, IVF are all examples. And we have three major markets. The research market, which is mostly drug discovery and drug development. Bioprocessing and production is the second one, and then medicine the third. And we're getting pulled into those very quickly. So I mentioned imaging, and this is an example of where we're orders of magnitude better. So what's different is we produce comparable images across time. You can even follow a single cell over time. We therefore can do time series over months where one can really study biology, which is a process. And we do this taking thousands of images. Within minutes, we take gigabytes of data. This is many orders of magnitude greater than what is done in those few labs that have some automation. And we take all these images with at least 100 focal planes, 2 to 50 microns apart. What that allows us to do, what's important about those focal planes is that we can image organoids, spheroids, tissues, organs on a chip. 3D cell culture is the fastest growing area within cell culture because it's a better model. The FDA Modernization Act 2.0 from last year, you can now enter trials without animal data, which is putting more pressure on cell culture. So we're focusing on expanding the market, and we're doing that because of the fact that we have all these different segments. We're starting off focusing on infectious disease, drug screening, stem cells, and quality. I have to add IVF as of our order from a few weeks ago. And our, actually now about 75% of our users with that order are using AI to make decisions. Prior to that, it was about 20%. The vast majority are commercial. I call it biology for business. And I've had this slide, actually showed it last time at IvyFON. I mentioned here that 10 customers plan to purchase an average of 25 instruments each. And in fact, that's what just happened with that order of 55. So I wanted to show you some images. Here are some of the key applications. These are images of organoids. This is from the Broad Institute. This is a remarkable slide. This was done by a drug company in conjunction with an academic institution. And for the first time, they were able to screen dozens of drugs with comparable data. Basically, that's a scratch. It's called a scratch assay. And those curves are showing how much of the scratch is being filled in, which is used for studying wound healing drugs. Previously, you weren't able to screen that many and you didn't have good data because people were estimating it, and people are very bad at estimating this. These are images from our customer who said Thrive will change the world. They wanted to be able to study SARS-CoV-2 without having to touch the cells again. After they put a virus on top of the cells, they stain them and count the plaques. Our images were so good they didn't need to stain. They also didn't need to even wait the required amount of time. At day 4 and 5, they were able to see the plaques. And at day 3, using some of our AI tools, we're able to tell them what will be the result. This is an example from Harvard Stem Cell Institute, where over time, we're actually able to follow a single cell. To know the origin of a cell, or to track the process for perhaps screening a drug to see the effect of a compound on a cell has been a holy grail that we're the first to meet. So our products are patentable, and to solve the kinds of problems that we are really requires cross-disciplinary approach. As I mentioned, we have 86 patent applications in 39 patent families, and we've had 38 patents issued, and they're in cell culture, stem cell culture, tissue culture, Internet of Things, collecting all the information in a cell culture lab, image processing, machine learning. So we have 20 employees. We were founded in 2014. So I mentioned my own background. I've started many companies, 7 of which are unicorns. My co-founder is the much smarter of the 2 of us. He's from Caltech, and he's responsible for some of the first sequencers when he was in Lee Hood's lab, and for many of the commercial ones, including Illumina's. We have on the board Mike Finney, who's a private investor and very involved with Life Science Angels, and Brock Reeve, who just left to start a fund, but was at Harvard Stem Cell Institute. Very committed to regenerative medicine because his brother, Christopher Reeve, was actually having very good success with stem cell therapies. He did not die from those therapies. So we've raised $35.4 million to date, mostly from family offices, such as the Estee Lauder family, KP Chow family in Hong Kong, Milstein family in New York, the Barton family in Massachusetts, the family that started Parker Brothers and well known for Monopoly. They sold to General Mills, and they're an investor in us, so we're spending Monopoly money, which as a kid I always wanted to see. So we're finishing up a convertible note. We're actually in our pro rata. I told Marty that I would make some available for people who are part of IvyFON. We've met a lot of our investors here. It converts at a 30% discount, and if you look at that discount, even with a quite high valuation, it will, with that discount, perhaps be a $45 million valuation based on some early term sheets that we have. So in summary, we're trying to transform the cell-based experiments in cell culture, which has been ignored in the past. We're bringing to cell biology basically what sequencing brought to genomics. Before sequencing, genomics had very little data. It completely transformed the field. And that's what we'll do with live cell biology, which is this island that lacks automation, data, and analytics. What we're trying to do here as well is to speed drug discovery, reduce that reproducibility crisis due to all the different areas, errors that are made, and the manual processes. And like the earlier quote, it's always good to hear from people who have used the system. Some examples are remarkable resolution. We see things we've never been able to see before. Tracking single cells and colonies in 3D is game-changing for drug discovery. This is what cell culture and cell imaging should look like. So thank you very much. And if there are any questions, I'd be glad to take them. Yes.
You sell the devices, but is there any razor blade to the business?
So the razor blades will come with our large full walkaway automation system, where most of our recurring revenues before that will come from our software. So as I mentioned, 9 out of our 10 engineers are working on software modules that we're beginning to sell this year. And those will be sold on a subscription basis.
And what kind of intellectual property protection do you have?
So we've really covered the field. We have a time, we hired a general counsel from one of the major law firm, IP firms that we use. We can afford him because he takes equity. And with our 86 patents, they're in image processing, machine learning, data exchange, ways of imaging cells, moving cells, manipulating cells. It's really a variety of hardware, software, security, because there have been many cases within life sciences where data have been modified. So it gives us traceability. Being able to track a single cell, to take the gigabytes of images we take in minutes, really requires a lot of expertise, which has been patentable.
And you don't see any competition on the horizon?
There's always competition. I really dislike it when a company says there isn't. The main competition is from some scanners that exist and microscopes. We're next generation microscope. We are doing what microscope companies should have done and did not do at all. They should have gotten better and better at capturing more and more data, huge data sets like we do that are comparable, but they didn't. And there's quite a bit of distance between them and us, and we don't cost that much more. We've actually been raising our prices over the last year and a half, and probably will continue to because we're meeting resistance. There are 2% of the labs do have automated scanners. They're actually for a different purpose, for medium-term and long-term assays, and their images aren't very good. They're actually trying to capture different kinds of data. Yes.
You said you raised 35 million to date. So what's your cash position now and what's your burn?
So our cash position is about $1 million, but we're planning on closing on over $1 million in the next week. And our burn rate is about $300,000 per month. It's been that for 4 years. We try to keep that low.
What's the biggest obstacle to adaptation from the medical community, from the lab community?
Good question. What is the major obstacle from labs and medical community?
Or you could address the sales, what's the length of the sales cycle?
I know. So the sales cycle, what we modeled turns out to be very different than what we're experiencing. We thought we would sell 1 or 2 to a lab, but in fact, we have all these opportunities where we're selling multiple, 25 to 100 instruments. A lot of the barriers to entry are it does take time for somebody to incorporate it into an experiment. So if they've been working on something, a field for years, they often don't want new data, but it's a very different. But the fact is, academic world and drug discovery is so competitive that they really don't have a choice.
I mean, it's actually pretty revolutionary what you're doing because, there's the world before of this, kind of handmade data analysis. And then you've got yours, which kind of steps in and quantifies and automates a lot of that, makes it uniform. So you don't have to worry about sampling errors, and then your methodology becomes pervasive, et cetera. So,
we were truly, this field, even though it's so important, has been in the dark ages. We're dragging it into the 21st century.
No, it's a big problem in it's opportunity in society that people don't realize that there's so much more data now, and you can make it clean and reach real conclusions. Right. So
an undigitized area is hard to find these days.
Fantastic, great job. Thanks so much.